good pronunciation. So, hello everyone. So I'm Mathieu Bolteau, a PhD student in the Digital Science Lab of Nantes, and I'm going to talk about the modeling of human embryonic development using Boolean networks as a framework. So, first, why do we want to model the human embryonic development? Because we need to better understand the human embryonic development in order to, in the future, improve the success rates of in vitro fertilization. But research on human embryos is very limited due to numerous factors, such as ethical or experimental one. That's why we want to have an in silico predictive model of the human embryonic developments. And few words about how works the human embryonic development. First, I'm focused only on the first day of this development, before the implantation of the embryo to the endometrium. And briefly, from the fertilization at day 0, the embryo goes through different developmental stages where different cell types appear that are represented in a different color here. And one main question for this presentation is how we can go from the medium trophectoderm developmental stage to the late trophectoderm stage. So concerning the data I have some single cell and realistic data from multiple stage embryos. And more specifically, I have the expression of more than 20,000 genes in approximately 1,700 cells from different embryos. And a previous study analyzed this data, and this study showed the different results. The first one is a clustering of cells, as you can see here with the UMAP. And they also identify some gene modules, and they characterize the pseudo-time evolution of cells at different developmental stage. But some words about the state of the art concerning the modeling of single cell data. So we identify 3 different types of methods. The first one, search to analyze the data using, for example, statistics like WGCNA, or machine learning techniques such as the UMAP. The second types of method consist to infer some networks, such as using correlation in order to infer some gene regulatory network, for example. But we can go further, and we can model the biological systems. And for that, in the context of single cell data and embryo development, we identify 2 principal studies. The first one consists to infer some dynamic Boolean models, but this method requires an average of gene expression and prior knowledge. And the second method consists to infer mouse embryo development computational models, but this method requires genetic perturbations and some knockdown on the system. So here, our objective for this project is to infer some Boolean models of different embryonic developmental stages. And for that, 3 challenges are present. The first one concerning the specificity of single cell data. In fact, single cell data are very sparse. They contain a lot of redundancy. We work with high-dimensional data. And the last challenge is the fact that we don't have access to any perturbation for this system. That's why we propose this type of solution, which allows us to distinguish between 2 developmental stages and build some families of networks models for each stage. And our method allows us to identify some regulatory mechanisms that differentiate both model and representing multiple cells. And for the moment, we apply our method on the medium and late trophectoderm developmental stages. So our method consists of a pipeline containing three steps. The first one is the Pekin construction, Pekin for prior clonage network. And the second step consists to construct an experimental design, and then we combine our two outputs to infer some families of Boolean networks. So the idea here is to learn some predictive models, and for that we have a prior knowledge network containing synced and directed causal interaction among genes, and we have also an experimental design which contains some gene expression for a specific developmental stage. And in this experimental design, we have some pseudo perturbation for different cells. And our pseudo perturbation I divided in two parts, the entries and the outputs. And the idea is to combine the PKN and the experimental design to understand how we can go from the entries to the outputs using a predictive model. So the first steps, the PKN reconstruction is based on the queries on pathway command database using a pbravo tool. And this tool allows us to reconstruct a PKN from an input gene list. And this output PKN is a labeled and oriented graph where nodes are genes or protein complexes, and the edges are the transcription regulation, such as activation or inhibition between some genes. And we also denote three types of genes in function of the topology of the graph. So in green, we have the input genes, which are not without predecessors. In blue, the readout genes, which are nodes without successors, and the other nodes in red are called intermediate genes. And here, outputs and intermediate genes are the entry for the experimental design for the second steps, and the readouts are the output for this second step. Concerning the second steps, the idea is to extract some pseudo-perturbation experiments from single-cell RNA-seq data, given the PKN structure from the step one. So before that, we need to preprocess our expression matrix, and for that, we reduce it, and we binarize the inputs and intermediate genes identified before in the PKN, and we normalize the readout genes. So if we take a toy example, and from the big initial matrix, we obtain this type of little matrix containing eight genes and 3 cells for the two classes A and B. And here the idea is to select K gene using a logic program in on-set programming. And here we select 3 genes, K equals 3, and the genes are A, C, and D. And the idea is to find some matching cells that are cells with the same Boolean vector for the K selected gene between the two classes. So for example, here we have 2 matching cells. The pair 1, 5, where we can observe the same Boolean vector for these two cells. And we have also the pair 2, 4. So these matching cells are also called pseudo-perturbation. And with the program, we force to have different pseudo-perturbation vector. Moreover, we have also a maximization steps in this program, and we search an optimal number of matching cells. And here, this number is equal to 2. Then, if we see the matching cell as a graph where cells are where nodes are cells and edges are matches, we can observe that we have here, for this example, redundant cells, and here the cell 1 and the cell 3. So these cells have the same Boolean vector for the k-selected genes. So we have two solutions for our problem. The first one containing the matching cells 1, 5, and 2, 4. And the second solution containing the matching cells 3, 5, and 2, 4. We also denote a score or a cell representativity, where for the class A, we have for the two cells that are selected, a representativity of 100% for the total number of cells in the class A. And for the class B, the two cells 5 and 4 represent 66% of the total cells in the class B. Then we search to maximize the readout difference in order to select the best cells in the redundant cells. And here, for this example, with this type of calculation, the program will select the cell number 3 for our example. To finish, the last steps concerns the inference on Boolean network using Casco. And the idea is to combine the experimental design and the PKN seen previously to infer some families of Boolean networks. And each families are specific for each class. And our Boolean network are both compatible with the topology of the PKN and the expression present in the experimental design. So regarding the results, first I show you here the reconstructed PKN from the input gene list containing 438 transcription factors. And we can observe this PKN have 233 nodes and 369 edges. Have a look now about the pseudo perturbation search. And here we fix k equal 10. And that means we search to select 10 genes for the 121 input and intermediate genes from the PKN. And here we have a combinatorial problem with a huge complexity. And regarding the result here, so in function, the execution time of the program, we can observe the number of pseudo perturbation. And we can conclude that we have a convergence of the number of pseudo perturbation over time. And if we focus more specifically on the best suboptimal solution containing 96 pseudo perturbation, we can observe that we have two solutions. So in other words, we have two possible subsets of k gene in order to obtain 96 pseudo perturbation. And if we look more in detail in this solution, we can observe 11 genes, and to conclude, we have 11 characteristic genes to have the same Boolean behavior in the two classes. Regarding the cell representativity, and more specifically for the solution 1 and the medium T stages, we can then see that our 96 perturbation represents 76% of total number of cells for these classes. And on average, we have 73% of representativity of the total number of cells for each stage. And the last result is the most important, the inferred Boolean network, and here for the solution 1. And we can observe a greater Boolean network variability for late reflectoderm stage, which can allow us to say that we have a gain of function for this stage. And this stage also seems more unstable regarding the number of Boolean networks. that can be seen as a transition from this late surfactordem to another one required to the embryo for develop. To conclude, I introduce you two main contributions. The first, which is an efficient algorithm and generates some robust solution. We discover 11 genes with the same expression vectors for different class across two classes. And these 11 genes are representative of the cell population. And more in general, we implement a method that learns Boolean networks for two stages using single-cell RNA-seq data and PKN. We identify some mechanism of gene regulation, and overall approach achieves a good computational time. And most generally, our method complements the state-of-the-art. So to finish, I would like to thank my advisors, Jeremy Carreto and Laurent, and I would like to thank you for your attention.
So in these two stages that you do, I don't understand why do you infer the Boolean network before essentially matching with the second part that you do when you do the data-driven inference? Why don't you fit the Boolean model after you have the PKN and the inferred network together ensemble? Or I misunderstood that you told me that. So you infer the Boolean network on the PKN.
Yeah.
And then with the data, you also infer.
In the beginning, it's not really a Boolean network. Yeah, it's just
just a static.
Yeah, it's just static. And after, yeah, for infer Boolean networks using the method Casper, we need both data. Exactly.
We have another question.